I'm going to be talking about the process of Sanger sequencing. So it was first developed in 1977 by Frederick Sanger. He also achieved a Nobel Prize for his work on this process. It is a tool designed for sequencing DNA, and hopefully the following animation will clearly describe the original basis of the technique and how it works. Firstly, the DNA that is to be sequenced is amplified through PCR. Heat is then applied to denature the DNA section and to separate the two strands of the double helix. This leaves us with a template or coding strand and a non-coding strand. We now see the primer that has been designed to affix to a specific DNA sequence of our template strand. It attaches to the strand's 3' end. Next, this template strand/primer complex is added to each of the 4 reaction vessels. Following this, DNA polymerase is also added. This polymerase will be used to anneal the deoxyribonucleotides to the template. In order to do this, volumes of all 4 DNTPs are added as well. Also included is the 4 different types of dideoxynucleotides. These are responsible for inhibiting chain elongation and for giving us sequences of varying lengths. Only one type of each DDNTP is included in each reaction vessel. To summarize, at this point, each of the reaction vessels will contain a template strand with primer annealed, DNA polymerase, DNTPs, and one type of DDNTP. The DNA polymerase allows the extension of the complementary strand in the 3' to 5' direction, starting at the primer. It will continue to add DNTPs until it reaches a DDNTP, which will terminate the sequence. The end result of this is sequences of DNA of varying lengths in all 4 reaction vessels. To actually sequence the DNA, the reaction mixture is loaded into a polyacrylonitrile gel electrophoresis plate. A power source is then applied, and as DNA is negatively charged, it will migrate through the plate from the negative to the positive pole. It is separated out according to the length of the DNA, as lighter, smaller lengths will travel further through the gel. In this way, we can read off the sequence by reading off the bands from the bottom upwards. The end result is a complementary sequence to the DNA sample from the beginning.